Hello everyone. So today we will be understanding label command. How we can label things in PyMOL, how we can label residues, how we can label atoms, how we can label chains. All those things will be covered in this video. Before we start this video, I want to thank you for appreciating all the videos on PyMOL. I have tried my best to create those videos and you guys really supported the content. Thank you so much. I have also created a video on show command. So please watch that video before watching this one, because you need to know how to show various things before you start labeling them, right? So in this video, what I'll be speaking, I will be talking about label command, how we can use label command in PyMOL and see various things. All right, so we'll start with the syntax, command syntax first. Let's move ahead. And this is the command syntax. As you can clearly see, it will contain the keyword which is label. So you have to type label in the command line first, and then after giving space, you can provide the selection. What is selection in this case? Selection specifies the atoms or residues to be labeled. So in this case, you will be providing the maybe number of the residues or specific atoms in this case. For example, from 100 to 200 that you can specify, or you can type all to select all the residues or all the atoms that are there in your structure. After that, you will be providing comma sign, and then after the comma sign, you will be having the expression. Expression in this case is the specific atom name, residue number, chain identifier, etc. I'm sure that when I try all those commands, you should be able to understand what I'm talking about, right? But in general, you need to be understanding that label command works by using the keyword which is label, after that space, then selection. In selection, you will be providing that what you want to select and what you want to label specifically. After that, the expression will be used by using the comma sign, and then space expression. Expression will be that whether you are typing atom name, whether you are typing residues and all. And we have the keywords for all of them. So let's understand some basic examples first. For example, let's see this command. In this case, what is happening? That it says label all, comma, name. If you directly type this, and you already have a crystal structure in PyMOL, if you just simply copy this one and just paste on the command line, what will happen in this case? It is going to label each. It is going to label each atom with its name. So the next example, let's move on to the next one, which is label all resi. So in this case, RESEI, what we are doing is we are typing label space all, comma, space resi. What this command is going to do is going to label each residue with its residue number. So in this case, resi represents the residue number. So all the amino acids, they are the residues, and whatever number they have, it will be printed out on the screen. Next, if you want to convert this to the chain, so what simply you should be doing is label space all, comma, space chain. So in this case, you will be providing the chain as the keyword, and this command will label each chain in the structure with its chain identifier. For example, you have chain A, B, C, everything will be labeled in detail. Okay, now let's go a little bit on the customized side of this command. As you can see right over here, what is happening, that label resi 20 to 30, and again we are providing comma sign and resi plus resin. So in this case, what will happen, it will label residues 20 to 30 with both residue names as well as residue number. So in this case, residue name will be printed out. In this case, the residue number will be printed out. So as you can see, we are also providing the selection in this case, 20 to 30. So this is how the basic command syntax work. So if you want to label everything with the number and the name, so what you can do is simply label space all, comma, space resin plus resi. So in this case, what will happen, it will label all the amino acids. It will specifically label the residues with the residue name and the residue number, and it will provide that in a single label, because you are combining these two things together, right? So all those things will be covered in this particular video before we move on to the hands-on session, because after this presentation, I'll be opening PyMOL. So I want you to also, if you can, practice all those things with me. This is going to help you to understand the commands better. So you can probably type all those keywords, what I'm going to show you in your PyMOL session, and see the output by yourself. With this, you should be able to understand how these commands, they work. And another thing is, we do labeling all the time in PyMOL. This is one of the most important skill set that you want to have. You need to know how you can label different things, how you can label residues, atoms in your PyMOL sessions, because without those labels, you are not going to have a publication quality images. Later on, I will also show you how you can customize the labels, how you can make your labels more beautiful, more appealing. What are the commands that you can use to beautify the labels? That will also be covered in this one. So this session is going to be really important for those who are beginner of PyMOL and who are just using the PyMOL for the first time. And if you are even experienced researcher and you're doing various things with PyMOL, maybe it's going to give you some additional information that you can use in your research. Okay, so with that note, I'll just complete this presentation part and we will move on to the hands-on tutorial part. Okay, so before that, I have one quick request to make. If you are new to the channel, then please do subscribe to the channel, because this gives us huge motivation to create more content like this and help all the people who are trying to use bioinformatics tools, or they are trying to understand the different types of topics in biosciences typically. Okay, so with that note, I will just conclude this session and let's move on to the hands-on tutorial part. All right, everyone, so let's get started. We'll start with getting the structure into the PyMOL software. And for that, we will be using command which is fetch, fetch space the name of the ID, protein ID in this case, which is 1H0M. And if you press enter, immediately the structure, and that was very quick, immediately the structure will be available to you and you can see the structure. This is the structure. Now, before doing anything in PyMOL, you need to study the structure. You need to see which structure you are using, and that detailed information, you can get that from the publication. Okay. And you want to study the structure in detail so that you understand what is going on, how it functions, what are the different aspects in this particular structure. So I know that this is the structure of TRA receptor, which is having a ligand molecule, as well as the entire structure is bound to the DNA molecule. So this is the DNA that you're seeing, beautiful structure. And then somewhere in the center, you will see ligand molecule. Now this, as you can see, you have two molecules, they are attached. I mean, it is difficult to see. So if I can just change the view. So what I'll do is, let's do preset in this case. So let's do preset and let's do pretty. So as you can see, different colored structures are visible, and you can clearly see you have orange, you have yellow, you have green and sand, blue. All these are same molecule. So they are just a similar molecule because it got crystallized. So all the molecules that are visible to you. So as you can see, you have different molecules. I mean, they're same molecule, but when it got crystallized, so you have multiple different molecules all together. So you can delete these molecules and just play with single protein in this case. So what I'll do is I can select these proteins by going to the selection panel on the right bottom of this panel, and if I click right over here, I can select chains. So what I'll do is let's delete or refine this structure a little bit, right? So in this case, otherwise it would be very difficult for us to understand the labeling command. Okay, so what I'll do is I'll just select all these chains and I'll just keep one chain. In case of DNA also, you will see you can select these extra duplicated molecules. Okay, so after doing this, I can go to A and delete or remove atoms. Now atom got deleted, and I'm going to remove these like ligand molecules also. So how you can do this by selecting and just removing them. Now, as I can see, what I have is one protein and one DNA molecule and one ligand molecule in the center, which is green in color. I can just zoom in and show you this. Okay, here it is, just sitting nicely in the ligand binding pocket. Beautiful. Okay, so this here we will only focus on the label command. We will not play around with the structure too much. Otherwise, this video is going to be very long video. So we'll directly start with labeling things. So let's see, this is the structure we have, right? And when you label, you want to see the structure in mostly in the, let's say in what format, mostly either in the line format or either in the stick format, so that you can see what you are labeling, especially you are going to label amino acids or atoms, right? So let's convert this entire structure and show this structure, and you can use show command, right? Why don't you guys use show command and see the structure here? I'm just going to use this panel and then just see everything as stick. I know it is very crowded, so we need to actually hide the cartoon. Okay, still very crowded, but I think let's move to hide stick, and let's work with show lines. Probably in that case, we will not be very confused or too much confused with the structure. So as you can see, this is the ligand binding domain. This is, as you can see, DNA binding domain, which is in light blue color or cyan color. In this case, you have dark blue, which is ligand binding domain. And in this case, the orange color, it is the DNA molecule, right? Okay, so let's use the label command. And I hope you remember that the first one that we understood is label, label space all comma name. So in this case, I'm sure that you remembered everything is going to get labeled with the atom name. So when I press enter, look at this. If I zoom in, probably what we can do is we can go to our ligand molecule, which is in green color. So you can see oxygen 36. So this is very important when you're trying to understand the interaction of atoms with other residues, or the other atoms in the different residues. So this tells you the identification of the specific atom, and it's very convenient. But in this case, as you can see, it's a little bit too crowded. Correct? But it's very clear. It can give you ID of every single atom. So you can map everything out. This could be important when you are trying to just understand which residue is interacting with which residue and which atom is interacting with which atom. Probably you are not going to label these things, but maybe when you want to go in very detailed analysis of the structure, probably then you will use that. Okay, so next is we are going to hide everything. So here, although I'm going to make another video on hide, but I'm going to use the hide command for this. Hide labels, and then labels, boom, they are gone. Okay, now next command that we have understood was the second one. I hope you remember. The second one was label, label, all, comma, and then we have resi. If you enter, so you can actually see that it gives you the ID or the number of residue. So you are getting the number of that specific residue. In this case, let's zoom in, and probably it's too crowded, right? Yeah. So it's giving you, in this case, let's understand the number right over here, and probably you're seeing a specific number is getting repeated. So this is 171, this is 170, and then 171. So this 171 is the residue, and then you can understand from where this residue is starting and from there where this residue is ending. And this you can use to identify to particular residue, and from where the starting point of the residue is and from where the ending point of that particular residue is. It's really useful to understand the structure, right? So this is, and I'm going to again use the hide command to hide everything, otherwise we will be confused. Okay, so before that, why don't I just change the appearance of the labels? Probably that will help us to see the labels accurately. So what I can do is, I don't know whether if I apply this command and it's going to be there all the time when we'll show the label, but I'm going to customize the labels right in the beginning. So what I'll do is I will be, let's say, use this label all label space all comma space resi. Okay, sometimes I'm not going to say space because it becomes so repetitive. So please understand by yourself. Okay, in this case, what I will be doing is I will be using the set command, set space label and then underscore size. So in this case, I'm trying to set the size of the label, and you can use comma after this and again space. Let's do 30, enter. Suddenly you can actually see the size of the residues or the labels of specific residues increase. So now we can probably see the number more clearly. Okay, you can also do this, which is setting the label color. So if you set and use space label label space, not space, underscore color, and then comma, in this case, which color? Yellow, probably. Yeah, so you can see, and then you can type whatever color you want, and then it should be highlighted right over here. So we have yellow labels, and I'm not a big fan of yellow color, but anyways, can we use any other color, please? Can we use, I don't know, light blue? Yeah, it's going to be very similar to the structure. I don't know. Okay, so let's use definitely not red, green, right? Light green. Okay, No, it's not taking. Light green, maybe spelling was wrong. L-I-G-H-T, light green. No, it's not taking it. Let's do green. Okay, green is fine, right? All right, so we can also change the font of this. So we'll do label, and then we will, sorry, in this case, first set space label font underscore, sorry. So it will be set space label underscore font underscore ID, comma. And there are different IDs. You can go to, a PyMOL website, and you can find out which ID corresponds to which kind of font. I've been using 7. So in this case, as you can see, it's a little bit bolder. You can use 6. Let's see, some Italian kind of font is also available. This was 6. Let's do 5. This is 5. Not much change. 4. Okay, 4 is not available. 5 is default. And then let's 8. Setting label font ID set to 8. Okay, so I was using 7. In this case, so it's been set to 7. I don't think so, right? Okay, so let's see the font as ID. Let's do, I was using, it's not changing too much. Okay, let's keep it that way. But this is the way you can change, and you can use different IDs. By default was 5, I guess. Okay, so now we have these customized labels, and you can do customization on these different labels. I'm still, thinking about this green color. Anyways, wow, it looked too crowded. What should I do in this case? Okay. Can't do anything, right? Let's decrease the size a little bit. So if you remember, what we have done is set label underscore size. And it was 30. Let's do 20, and yeah, that should be good. Otherwise it was too big. And if you zoom in, zoom out, the size will probably change. So you can use the font, and you will have color different in the case of if you have changed the label, right? All right, let's go next. We have used how to label the residues. Now next command is we will be using chains. So before that, we will be hiding. So we'll do hide space labels is hidden. Now we want to label the chains, and we'll see how the chain labeling is done. Label chain. So for example, if I want to label the chain A, so I will be typing A in this case. If I have multiple chains, I don't know. I have deleted so many chains, but yeah, I have not deleted A. So as you can see, now every molecule will show you this is chain A, right? And then if you have chain B, that will be highlighted. So this is how you can also identify which one is what. Okay, so let's hide this one. I hope now you understood how the specific command work, which is label chain A. All right, next is label. Okay, so in this case, we will combine the number of a particular residue with its name. So we'll be doing label space resi 1 to 15. Now just not to see too many labels, I'm selecting a part of the protein from 1 amino acid to 50. I don't know whether we have those amino acids right over here. So how you can check is you can click on sequence, and then you can see we have from 1 to 50. So we're going to label all those. So label space resi 1 to 50. So we are selecting the residues from 1 to 50, and after that we will be typing comma resin. So we want to have the residue number and the specific residue here. There you go. Look at this. So in this case, what we have done is we have selected the specific residues, and those residues, they are highlighted with their specific number, right? So you can see, yeah, let me zoom in. So this is glycine 24. This is also glycine 24. This is isoleucine 14. Leucine 21. So you can see number is there. Also the ID of that particular residue is also there. So it's super helpful to actually get the information immediately about this. So whatever you have selected, that will be highlighted. Okay, let's hide this one and let's go further. I'm going to just repeat the hide command. And next in this case is a little bit advanced command. We will be doing this. Okay, the next command is going to label only the backbone atoms. Backbone atoms, especially the nitrogen, and then you have carbon, alpha carbons, and combination of residue as well as the residue name and the number. and also the atom name. So it's going to be a combined command. How we can do this? So we have to do a label and then space. We'll start with bracket and we'll say name, because we want to have name of N, we want to have name of CA, we want to have name of C. So we'll close the bracket with comma. We also want to have resin, residue number, and then space. We want to have a residue also. Then we'll use the plus sign. Okay. After this, we will also use the dash sign under the inverted commas and plus name. So all those things together will give us the detailed information on a specific residues that we want, since we have selected the entire molecule. So what we are getting is, you can clearly see. So when we are putting this dash sign, so that dash sign will appear because we are putting it inside the inverted commas. It's a typical Python syntax, right? So as you can see, what you are getting is, this is the information. Look how useful this information is. So you are getting the residue number. So let's take the example of anything like serine, maybe right over here. Yeah. So this is the nitrogen of serine 160. This is the carbon alpha for serine 160. So you exactly know which atom or which residue you are trying to identify. This is helpful. I'm again telling you, when there are interaction happening or you want to identify distances, so you can exactly pinpoint which molecule is interacting with which molecule. So it's super helpful command that you probably will be using. Okay, let's again hide this. Okay, so I'm sure now you have already memorized that hide labels is going to hide everything. Okay, starting with another command. This command is going to only label the chain A residues, combining the residue name and the chain identifier. So if you sometimes you want to identify residue with a chain identifier, so what you can do is label space and then use the bracket sign, chain space A, close the bracket, and then use comma space RESN residue number, and then again I'm just putting inverted commas because I want to have that slash sign. Okay, in this case we are not using slash sign. I forgot that. So plus sign, and then you will use inverted commas. In this case we will be typing chain keyword is there, so we will see the chain keyword will be written plus the chain, so that we know which chain we are using. Okay, enter. Look at this. So we want the dash, that is why we are using dash chain in our syntax. And then in this case you can clearly see isoleucine chain A, right? So this is the part of the chain A, this is the part of the chain B, because we are just highlighting chain A, so you should only be seeing chain A in this case, not the other residues. Yeah, look at this. So this is our entire chain A. Other chains I've deleted. So you can zoom in and you can see arginine, valine, everything is labeled nicely. Okay, now let's move on to a little bit more advanced command, and I'll just go and hide everything. Okay, in this advanced command, what I will be using, and you'll see, and I want you to also understand by yourself. So what I'm doing is label chain A. Okay, so I hope that is clear. And I'm using AND operator in this case, resi 50 to 60, 50-60. I hope now that is also clear. We'll use comma RESN. I hope that is also clear that we are using residues. Now I'm going to use slash sign. That is going to add the slash sign, and this just concatenating the strings, and then we'll use resi. Okay, so when you do this, you will see this is going to highlight a specific part of the protein, and let's do next also. Let's run this. Okay, so let's do second part, because whatever we have highlighted, I want to change the color of that one. As you can see, I have only highlighted the specific part. In this case, I've highlighted only the 50 to 60 part of that particular protein, and then I also want to change the color of this particular part, chain A and resi 50. Probably I should be using this in my another tutorial, how to change the color of specific part of the protein. Now look at this. If you see carefully, that I have changed the color of all the residues that I've labeled. And if I show you this because I've been creating all those things one after another. If I show you this, these are all colored, and if I show this structure as cartoon and hide, yeah, probably you should be able to see this very, very clearly now. So what I've done is specifically highlighted a part of the protein by selecting the residues and also change the color by simple command. Look. So maybe you want to highlight this region because there is a conformational change in this particular protein. So you can do this by using that specific command. Okay. And now let me show you another important, most of the time that I use this particular command all the time. For that I have to hide everything. So what I'll do is I'll just simply go to the command line and I'll hide labels. Okay. And then I will also hide, not hide, let me just apply this. Okay. So what I'll do is in this case, I want to see the active site residues. So what I'll do is I will hide. Okay. First, let me select the residue, which is not the residue, the ligand molecule. And when I select this, I need to change or create the selection. In this case, I'm typing ligand. So now you can see new ligand object is being created. And in this case, when I hide everything, nothing else, as you can see, ligand is selected. I want to show the ligand as, let's say, sticks. So ligand is there. Now, when you have created objects like this and you want to see whether there are, residues which are very, very close to this ligand molecule. So what we do in this case is we can use this simple command, and you all are going to love this command, which is show sticks. I'm going to now combine what we have learned so far by res, all the residues, all within, let's say, 5 of ligand. So what I'm doing is it is going to highlight all the residues that are within 5 angstrom of the ligand molecule. Look at this. By just typing this, I got all the residues that are closer to this ligand molecule, and that is my active site. Isn't it? So now if you want to label these things, so let's see if you want to, let's see, I don't know whether this is going to label the residues. It will obviously label all the residues, so you don't want to do that, right? Hide labels. So in this scenario, what you can do is, because you know these are the residues, and you can just click on residues, especially, and then select all the residues, specific residues that you want to label them. Let's say I've just selected few of them, not all of them. I don't have that time. Okay. So what I can do is again create another selection, which will be the active site. Active site. Okay. Sorry. So I have to redo this. There might be some other way to do this also, but this works and you have to be careful, right? So let me, because if you type somewhere in the black background, this is going to unselect everything. So let me, and let's go to selection, rename selection, and this is my active site. And if you already see, if you already know what is your active site, then there is no need to do this exercise. Okay, now next is you need to label the active site, and let's go to label and label the residues. Look at this. Now in this case, probably you need to customize your label, especially increase the size. So in this case, I'm going to set, I'm going to probably increase the size of the set label underscore size. Let's do 4040, and okay. Okay, you need to use not without set. So set space label underscore size, comma 40. Look at this. So now you can actually see the font size increased. So as you can see clearly that this is how you can label so many things in PyMOL. You can customize the labels, and there are so many other things you can do. If you have a basic idea of how Python syntax work, you can understand using PyMOL wiki how various commands they work, and I'm here to help you out also. You can post questions if you have any, and I've just shown you various commands and that you can use in your analysis of your own protein molecules, right? All right, so I'm going to stop this video. I hope this video is not too long. I still have so many commands that I can show to you. Maybe later on I'll make smaller videos on those, like how to show aromatic rings, aromatic amino acids, how you can show polar amino acids, how you can show combined polar, non-polar, so many things you can do. Just if you understand the basics, you should be able to do everything that we can do in PyMOL. It's really great, and do not forget to save the session, right? You can save the session and then you can come back and complete your analysis and have amazing images and amazing analysis done. Hopefully contribute to a really, really good project. Okay, so I'll just stop this video right over here. If you have any doubt, any questions, just please let me know. I'll be creating more videos and I'll be coming with more detailed, very, very important content in your future. So if you're new to the channel, then please do subscribe to the channel. If you have seen this video up until end, that means you like the video, and please hit the like button. Okay, so I'll meet you in the next one. Till then, take care, everyone.